Welcome to Quick MCAT Lessons, where I try to explain MCAT topics in 10 minutes or less. Today we're diving into the structural framework of the cell, the cytoskeleton. But this is more than just scaffolding. The cytoskeleton gives the cell its shape, enables it to move, helps transfer material, and plays a critical role in cell division. Now the cytoskeleton is made up of three main types of protein filaments: microfilaments, intermediate filaments, and microtubules. Each of these has a different structure and function, and all three work together to support the cell's dynamic needs. Let's start with microfilaments. These are the thinnest of the cytoskeletal elements, and they are made of a protein called actin. They're especially important for maintaining cell shape, forming the cortex just beneath the plasma membrane, and they are also key players in muscle contraction, where they interact with myosin motor proteins and get force by using ATP. Microfilaments are also involved in cell movement. This is when immune cells crawl through tissues. They're also used in cytokinesis, which is the final splitting of the cell during mitosis. During mitosis, the cleavage furrow is also formed. The cleavage furrow is a groove that appears in the cell membrane of dividing animal cells during cytokinesis. This is the final stage of cell division, and here the cytoplasmic organelles are separated to form two daughter cells. This groove is formed by a contractile ring composed of actin and myosin filaments, and this contracts to constrict the membrane and divide the cell. Next up, we have intermediate filaments. These have a rope-like structure and are more stable than other cytoskeletal components. They provide tensile strength, helping cells resist mechanical stress. You can think of them as internal cables that hold everything in place. Different types of cells have different intermediate filaments. For example, keratin is found in epithelial cells, where neurofilaments are found in neurons. The nuclear lamina, which supports the nuclear envelope, is also made of intermediate filaments called lamins. Now for the largest cytoskeletal element, microtubules. These are hollow tubes made of alpha and beta tubulin dimers. They radiate out from a central structure called the centrosome and are highly dynamic, which means they're constantly growing and shrinking. Microtubules act like highways inside the cell. Motor proteins, like kinesin and dynein, move along them, carrying vesicles, organelles, and even chromosomes during mitosis. Microtubules also form the core of cilia and flagella, which are structures used for cell movement and fluid transport. Cilia are projections from a cell that are primarily involved in movement of materials along the surface of a cell. Flagella are involved in the movement of the cell itself. They both have the same microtubule structure, with 9 pairs of microtubules forming the outer ring and 2 microtubules in the center. This is known as a 9+2 structure, and is only seen in eukaryotic organelles with motility. A 9+2 structure, so there are 9 pairs on the outside and 2 on the inside, so in total there are 20 microtubules. Now let's talk about centrioles. Centrioles are found in centrosomes, which are organizing centers for microtubules, and are structured as 9 triplets of microtubules with a hollow center. So just to recap, you have a microtubule with the 9+2 structure, and then if you take 3 of those, you get a triplet, and if you take 9 of those triplets and arrange them in a circle, you get a centriole. And a centriole is a MTOC, which is a microtubule organizing center. And during mitosis, the centrioles migrate to opposite poles and organize the mitotic spindle. The microtubules go from the centrioles to the chromosome and attach with the help of complexes called kinetochores. These pull apart sister chromatids. Now there are also some motor proteins that are essential for movement within the cell. You have kinesin, which typically moves cargo towards the plus end of microtubules, away from the centrosome, and you have dynein, which moves towards the minus end. This is towards the centrosome. And you have plus and minus ends because these are polar. So once again, minus end is toward the centrosome and the plus end is away from the centrosome. And the plus end is kinesin and the minus end is dynein. This directed movement is critical for processes like neurotransmitter transport in neurons or delivering vesicles to the cell membrane. Another factor of cell motility is microfilaments. Think about how a white blood cell crawls by extending its pseudopodia. This is driven by actin polymerization. So as I hope you can see by now, cytoskeletal components are very important, and defects in these can lead to a variety of diseases. For example, mutations in keratin can cause blistering disorders in the skin. Also, drugs like Taxol and colchicine target microtubules and are used in cancer therapy because they interfere with mitosis. Even neurodegenerative diseases like ALS have been linked to abnormalities in intermediate filaments. And just to wrap up, so the cytoskeleton is much more than a cell skeleton. It's a dynamic system that gives a cell shape, enables movement, organizes transport, and powers division. And that's it for this one. Next time we'll talk about tissues.